Good morning, everyone. I'm Christy Risk, Senior Editor at Genome Web, and I'll be your moderator for today. This is the first part of a three-part series called New Frontiers in Liquid Biopsy Research, sponsored by Thermo Fisher Scientific. The title of today's webinar is Adding CNVs and Fusions to a Lung cfDNA Assay, Impact on Oncology Clinical Research. Our panelists today are Dr. Bea Bellosillo, Head of the Molecular Biology Laboratory at the Pathology Department of Hospital del Mar and Associate Professor of the Pompeu Fabra University of Barcelona, and Dr. Kelly Bramlett, Director of R&D in the Clinical Next Generation Sequencing Division at Thermo Fisher. You may type in a question at any time during the webinar. You can do this through the control panel, which usually appears on the right side of your screen. Click on the Q&A box on the upper right side of the control panel. When you click on Send To, please select All Panelists. We will ask the panelists questions after the presentations have concluded. Dr. Bellosillo, please go ahead.
Thank you very much. Good afternoon here in Spain. Good morning to all of you. I thank very much the opportunity to be here today and share with you our experience working with liquid biopsy samples from individuals with lung cancer. First of all, for those of you who are not so familiar with lung cancer, let me please give a brief introduction of the role of molecular markers in this disease. I will then go on to plasma briefly for biomarker detection, and finally share with you our experience with the NGS applications in liquid biopsy. Briefly, just to remember that lung cancer is one of the leading causes of cancer-related mortality among men and women worldwide, with a 5-year overall survival rate of 16%. The therapeutic approach to individuals has changed very much in the last 25 years until the late 1990s. All cases with advanced lung cancer received the same treatment with platinum-based combination therapy, irrespective of their histological subtype. Later, treatment decisions were taken based on morphology first, and then in the histological classification of tumors. And it's been in the last 10 years that we have reached a better understanding of molecular abnormalities present in lung cancer that define disease subsets, but more importantly, we have developed directed therapies targeting specific gene alterations that cause the aberrant signaling and proliferation of these cells. These mutations, these alterations are mainly mutations in the EGFR gene, but also fusion genes like involving ALK, RET, and ROS genes. Currently, based on this knowledge, all patients undergo tissue biopsy, always for performing the diagnosis, the histological diagnosis, but also to perform molecular profiling of the tumors. Here you can see in this slide the most frequently involved genes in these pathologies, and these gene alterations have been included as part of the testing that has to be included in all cases before deciding which is the best therapies. Not also at the molecular level, also immunohistochemical testing of PD-L1 for deciding immunotherapy. This testing, and this is also an important point, and that's why we are all moving now to NGS, it's recommended that it should be conducted as part of a broad molecular profiling. This molecular profiling will not only be performed at the time of initiating the first-line therapy, but also when patients may eventually relapse at a known therapy, at a targeted therapy. And in this sense, I would highlight the mechanisms of resistance accounted in individuals that are receiving targeted therapy anti-EGFR, that is tyrosine kinase inhibitors, that after a certain time may develop resistance. One of the most frequent mechanisms of resistance is the acquisition of a T790M mutation. And the importance here is not only to know the mechanism of resistance, but also that the presence of this alteration opens the path to receive third-line therapies that have been already developed and are working fine. So normally, as I mentioned, all these biomarker detection is performed in a tissue biopsy. However, as you may already know, tissue is always an invasive procedure. It's not infrequent that the amount of material we have is scarce, is little, and this is especially true for lung cancer, where it's often that biopsies are extremely small. Another handicap of the tumoral tissue is what's known now as tumoral heterogeneity. That is that even within the same tumor, we may observe different molecular alterations depending on which area we are analyzing, depending on which area has been biopsied. Also, we have to have in mind that to have a biopsy, it's sometimes difficult to schedule in the routine day. And finally, one of the things we are seeing now is that we want to monitor our patients to see how they are responding for therapy. And of course, to perform sequential biopsies, it's not always recommended. For these reasons, nowadays, liquid biopsy appears as a good alternative or a good complement to molecular analysis in tumoral tissue. The reasons, of course, are, well, that it's minimally invasive, that we know that it can represent the global tumoral load, the global mutational profile of all the tumor and the metastases present, that it's easy to get dynamic information, so while we are following the patient, that it's easy to schedule the obtention of this type of samples, and that it looks like we are able to use it to either detect early responses, but also to detect earlier relapses. So for these reasons, it's now being incorporated as part of the routine as well as part of research aims in many institutions. As part of the molecular lab, one of the things that is very important for us is which technique should we select to analyze these samples. This is widely known cartoons showing the mutation detection sensitivity depending on the technique. Traditionally, as we know that mutations in plasma samples may be present in a very low percentage, we are selecting those techniques that may be most sensitive, like digital PCR or similar techniques such as beaming. However, we have the need always to move to a broader analysis using NGS. That wasn't possible several years ago, but since the development of the molecular barcoding and the incorporation of this approach in several kits like the ones developed by Thermo Fisher, we are now able to apply NGS, analyze a wide range of genes with a sensitivity of up to 0.1%, which makes it possible to apply it to plasma samples. Moving then into NGS, briefly to remind you how this molecular barcoding works. Normally, when we were applying conventional NGS targeted DNA sequencing, what we were doing was to either based on amplicons or capture, sequence a region a number of times. And when we got the results and saw a variant existing in the sample, if the variant was only present in a very small amount of reads, it was difficult to say whether this was a true variant or it was an artifact from the PCR amplification or the sequencing reactions. With the incorporation of the molecular barcoding, it is easier to discriminate between an artifact and a true variant. What we are going to do is to incorporate a small sequence of DNA at the very beginning, so we will independently label all the DNA molecules present in the sample. At the end, when we analyze the results, if one of variant is only present in one of all the reads that have the same molecular barcode, here in yellow, this would mean this variant is an artifact that has developed during the sequencing, whereas if we have this variant in all the reads that have the same barcode, the same tag, here in red at the right-hand side, this would mean that this is a true variant. And with this approach, we are able to increase this sensitivity down to this 0.1%. The first kits we tested were the kits that were aimed at analyzing mutations. This panel, the Oncomine Lung cfDNA assay, is targeting 11 genes that have a role in lung cancer, and it's aimed at analyzing specific spots. We applied this panel to different samples that we were working with at our institution, and just to show you a case here with sequential samples from a patient that had been receiving tyrosine kinase inhibitors. We had monitored this patient with digital PCR for his sensitivity mutation that is shown here in blue. You can see how the mutation was present in the initial sample, how it was undetectable after some time and then became detectable again when the patient started to progress. At the same time, also using digital PCR, we were able to detect the emergence of the mutation of resistance, the T790M mutation. And at the same time, we were able to get also a biopsy where the mutation of sensitivity was present, but not the mutation of resistance, showing this heterogeneity that we were talking about previously. And what we did was to apply these NGS panels at the first sample and at the last sample. And you can nicely see how we were able to detect the EGFR mutations, both of them at the time of relapse, with percentages very similar to those obtained by the digital PCR. And in addition, we were all able to detect a P53 pathogenic mutation that was not only present in the last sample, but also in the initial sample, suggesting that P53 was not responsible for the acquisition of resistance. It was only the T790M mutation. We also analyzed more samples from patients at the time of progression. Applying these kits, we had a total of 22 cases. From them, we could analyze 13 of them and saw that the T790M mutation was the most common mechanism of resistance, although with the NGS panels we were able to detect concomitant presence of P53 or other mutations in green, or only P53 RAS mutations here in purple. But even in one case, we didn't found any mechanism explaining the acquisition of resistance. Of note, I would like to highlight here that from the 22 cases, we were only able to perform the analysis in 13 of them. The reason was that in the remaining 9 cases, the amount of total circulating free DNA we obtained was not enough to be able to undergo NGS analysis. And I will go back on this issue later. So now moving on to the other kits that have been recently developed. It was about 1 year ago that we had the opportunity to start testing the lung cell-free total nucleic acid research assay. The idea was not only to cover single nucleotide variants, which were also including the 11 genes with the hotspots, but the idea was to obtain from the plasma samples DNA and RNA to be able to analyze single nucleotide variations, but also copy number variations, as well as the fusion genes. Copy number variations involving a MET, and the gene fusions involving ALK, RET, and ROS1. The idea is that here in this slide you can see the workflow. We will start working with the peripheral blood tubes. Here we've stated single blood tubes. That would depend on the type of tubes that the institution is using. It's important to have 4 ml of plasma, so in case the tubes are smaller, it should be 2 or 3 tubes. From here, and using a MacMax kit that simultaneously extracts in the same extraction both DNA and RNA. Afterwards, we will perform first step of retrotranscription with the supervelo to convert RNA into cDNA to enable then the detection of the gene fusions and the exon skipping of MET. After the retrotranscription, we will perform library preparation, then sequencing, ideally on the GenStudio S5, as it is more cost-effective to perform several samples at a time. And finally, the data are analyzed using the ION Reporter Cloud software. And just to show you our experience with it, we have chosen 2 cases where we used this kit and obtained very nice results. The first case is a case of a 69-year-old woman that had been diagnosed with lung adenocarcinoma. We obtained the tissue biopsy and looked for biomarkers for having the option of giving her targeted therapy, but there were no mutations in the EGFR and no gene fusions involving either ALK or ROS1. Also, the expression for PD-L1 was negative, so the patient had no option to targeted therapies, and she received standard chemotherapy. After a while, we saw clinical progression, and we obtained a liquid biopsy sample in which we were interested in looking for some additional biomarkers to offer her the participation in any clinical trial available. We took advantage of having access to this early access kit and analyzed the sample with it. And I'm not sure, this is the result we obtained. I'm not sure if you can see it properly or it's a little bit small, but if not, I will show you. We obtain in the first column we have the name of the sample, and the second column we have the detail of the single nucleotide variations and indels, where we found 2p53 mutations. In the third column we have copy number variations, that there were none detected, and in the last column we have the fusions, where you can see that there was a KIF-RET fusion present. This was great news because RET is one of the driver genes involved in lung cancer. Several mutations involving the RET gene have been described so far, but the most interesting point is that there are some targeted therapies available for these cases. As it was a research use only kit, what we did was present the results at the molecular tumor board, discuss it with the pathologist, and what we did was to go and look for the paraffin blocks, see if there was any material available. Luckily, there was material available, and we were able to perform FISH analysis on the sample, as well as the Oncomine solid tumor CIVD assay. And with both approaches, we confirmed the presence of the RET fusion in this patient. So with this, there was potential targeted therapy identified, and therefore the option to receive new therapy. And the second case I would share with you is a case of a 48-year-old woman, also diagnosed of lung adenocarcinoma, in which we had previously detected by Sanger sequencing deletion in exon 19 together with a mutation in exon 20. This mutation in exon 20 is not very clear whether it gives huge sensitivity to TKIs, but so she was given one of the second generation, afatinib. She responded for several months, but afterwards experienced a slow progression. At that time, we looked for mutations using liquid biopsy to try and detect the presence of the T790M mutations. We used ARMS real-time PCR, and that was negative for the T790M, positive for deletion in exon 19, which suggested that there was enough tumoral DNA present in the sample. Several months later, what we did was to analyze it with this NGS kit, and this time we were able to detect the deletion in exon 19 together with the presence of the T790M mutation and the p53 mutations. We confirmed the presence of the T790M with digital PCR, and therefore at this moment she has potential treatment with third generation TKIs. Some final considerations when thinking on applying NGS to plasma. I think it's extremely important to quantify the amount of cfDNA obtained from the sample and to try and work with as many milliliters of plasma as available as possible. We have to have in mind that the amount of cfDNA varies from sample to sample, from individual to individual, and even in the same individual, depending on the time of the disease, on the therapy he is receiving, this amount may vary enormously. So it's critical to perform an accurate quantification before starting doing any NGS application. Normally, what is recommended is to work with at least 20 nanograms of DNA if we aim at going down to a 0.1% limit of detection. Sometimes this amount is not possible to achieve. In that case, of course, it's worth going for it, but always having in mind that we will not be able to go this down in sensitivity if we are not working with these 20 nanograms. And finally, thinking on the clinical application of these results at some point for decisions in the clinical setting, it's also very important to have in mind that these results must always be discussed in multidisciplinary tumor boards to get the best decision. And with this, I want to thank Thermo. Thank you for your attention, and also thank all my colleagues from the pathology and the oncology department and people from Thermo Fisher that have helped us in setting all this work. And with this, I give you back, Christy, the voice. Thank you.
Thank you, Dr. Bellosillo. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. We will conduct the Q&A portion of the webinar after the presentations have concluded. Next up is Dr. Bramlett. Please go ahead.
Hi, everyone. Kelly Bramlett here. I am a director in research and development at Thermo Fisher Scientific. And for about the last 3 years, my team here in Austin, Texas, has been developing new methods, workflows, kits, analysis tools to be able to sequence rare variants from liquid biopsy samples using our Ion Torrent sequencing platforms. So really great talk by Dr. Bellosillo. And hopefully I'll follow up now with a little bit of our internal work that we put in to develop these kits and get them out broadly for you all to use and hopefully benefit individuals like you've just seen. So here's a slide just showing our whole portfolio right now of the Oncomine cell-free assays that we have. We have quite a few available. The most recent one is shown there on the left. That's our largest panel. It's a pan-cancer panel covering 52 genes and all the different variant classes that Bea just referred to. We have smaller panels that are more focused on lung, which we just heard a lot about. And breast cancer, we have a couple of options there, and then one for colon or more broadly GI types of tissues. So the whole idea when we were developing this portfolio is to create a technology that would allow us, will allow you all to detect variants down to 0.1%. And you just saw a nice description of what that means, right? There was a nice slide with the needle in the haystack, looking for a needle in a haystack. When you're thinking about liquid biopsies and trying to detect that very, minimal amount of circulating tumor nucleic acid, you really need a technology that will allow you to detect it down to one variant copy of nucleic acid in the background of about 1,000 copies of wild-type material. So that was really the design challenge that we had. We also wanted to do this from a single tube of blood. and wanted to really optimize our content for primary tumor drivers and resistance mutations, getting at the information that's going to be most valuable to the clinical researcher. So Bea had a nice slide about this, but I'll talk through it again a little bit more specifically to the Oncomine cell-free assays. We basically use a hybrid set of primers to target our specific areas of hotspot known and well-established hotspot mutations. And attached to those primers is the unique identifying sequence that you heard about. What this allows us to do is really monitor each fragment of cell-free DNA that's captured and group all of the amplified products that go with each starting molecule together so that we can analyze each amplification product together with its initiating molecule. That way, we're able to detect a very rare variant that might be present in the starting fragment that was hybridized to and captured in a library prep from the cell-free population. So as shown on the left here, if you do have a rare variant in your starting molecule, you'll see it in every amplified product that comes from that starting molecule. And that's kind of shown in the cartoon on the left. We actually require 2 unique molecules to be identified containing each variant that is reported back with our analysis solution. So that's one way we increase the specificity, really kind of getting at the high confidence in detecting these rare variants. Alternatively, if there is no variant in the initial material, you won't see a single nucleotide change in each amplification product as shown on the right. So this is really how we accomplish getting from the higher LODs that Bea showed from traditional methods of NGS sequencing and using some molecular biology and analysis tools and optimizations to drive that LOD down to 0.1%. This slide shows really two major things, and I'm going to start on the right because you've just gone through a nice slide about the math. So this is the way I like to talk about it. On the right, you can see we have a graph here that shows you how the input material that you have, the input amount of cfDNA on the right axis, really has a direct impact on your potential limit of detection with each sequencing run. And that's shown across the horizontal axis on the bottom here. In order to hit these limits of detection, we have the amount of sequencing coverage you need of each amplicon. That's shown on the left here. So as we just talked through, in order to really hit the target of 0.1% variant frequency, you need about 20 nanograms of cell-free DNA. Bea nicely walked through the math there. We're assuming that's around 5,000 to 6,000 copies of cfDNA, and we're requiring you to capture 2 of those in order to call a variant. So that's how that math works out. You can use less, as we have talked about, and I have down below this graph some specific amounts of cfDNA and what limit of detection we internally were able to get with 1 nanogram, 5 nanograms, 10 nanograms. So certainly you can use 10 nanograms of cfDNA, but your LOD changes a little bit to about 0.15%. So this is just a way to kind of get your brain around the math of how many molecules you need to put into library prep in order to see those variants and what level those variants can be at and you can detect with statistical significance. On the left, this table shows the results of some of our internal testing. So when we develop a new workflow or a new sequencing kit, we go through a series of testing. We call it verification and validation testing. This is only analytical, no clinical types of verification, but analytical verification, just making sure that what we've developed is working the way we expect. So what we do is take contrived material that has been fragmented to mimic the size profile of cell-free nucleic acid, and that material has engineered variants, expected variants in it. We make mixtures of those fragmented material into the background of well-understood, genetically evaluated background material. And we make mixtures where the variants are present at either 0.1% or 0.5%. And then we test across 4 different users and 2 different sites, and include a bunch of different sequencing instruments to try to incorporate as much kind of variation as we can into this testing. And what you see on the left is the results of our ability to call the expected variance, that's in sensitivity, and a measure of specificity, meaning did we call any false positives. So we have very high sensitivity and very high specificity. And that's just a result of these kind of technologies that we've talked through and the optimizations that have been made. The next slide. I show the whole portfolio again, and what I'm really going to concentrate on is, Bea talked nicely about our initial lung panel that really just targeted single nucleotide variants and hotspot indels. And we really wanted to, especially for lung, expand that content and include the fusions, the Alkret and Ross fusions, and also MET exon skipping, which is an important resistance mechanism to current targeted therapies. So we had a second version of our lung content that really kind of expanded our capabilities in different types of variant detection from liquid biopsies and the cell-free component. Bea nicely went through this, but I'll talk through it again. We really expanded from DNA only to capture both cell-free DNA and RNA. So we call that cell-free total nucleic acid. And we expanded from the hotspots to include CNVs and also these important fusion genes. So I'll talk through a little bit of our experience there and what you can expect from these additional variant types. As was pointed out earlier, Thermo Fisher launched a optimized cell-free total nucleic acid isolation kit. It's one of our MagMax product lines, and we used this extraction method during our internal analytical verification. So we're again talking about cell-free total nucleic acid. And another key change that was mentioned earlier is there is an RT step that's added prior to prior to the hybridization of the gene-specific primers. So after RT, you have in your mixture both DNA and cell-free cDNA, right? And the primer pool will sit down on its expected target, whether that is a DNA target or a cDNA target. Works very well. We had to optimize the designs here to make sure that all the different primers worked well with each other. But it was really a great new advancement in trying to get more information out of a single tube of blood and a single amount of nucleic acid. So here's in detail about the original content of our lung cfDNA assay shown on the left. and how we expanded that in the second version, our lung cfDNA assay. As was pointed out, we added the fusions to ALK, RET, and ROS that are targets for targeted therapies. We added the MET amplification, that is a resistance mechanism. We also have MET exon skipping assays here. And then we kept all the CNV content that was in the first panel. This new assay that we've been talking about today can really be run as a 6-plex, so combining 6 libraries together and running them on a IN 530 chip using the S5 system, or a 24-plex on the larger chip type, which is the IN 540. So in order to, do some internal testing around these new variant classes, we used new types of positive control material, right? So I talked to you about the contrived material we used for CNVs and indels, and those results are shown here again for reference. And now we've added on really three classes of new kinds of variants. We kind of combine the MET exon skipping and fusions together in our report, which I'll show you later. That's because we detect both of these in the RNA space. So we kind of think of these together. But we did have to generate some new control materials for MET CNV. We actually got some cell lines that harbored a known MET amplification, and we isolated cell-free DNA from the conditioned media from that cell line. And we titrated that into the background of healthy donor cfDNA to create a positive control for MET CNV. Same kind of concept for fusions. We have cell lines that harbor known ALK, RET, and ROS fusions, so we tested a fusion variant of each class from each driver gene. And same with MET exon skipping. Again, a cell line harboring MET exon skipping. So we created new types of contrived material here to measure our ability to detect these expected variants and also evaluate any false positives. So the results are shown here. Our mixtures that we made and tested contained fusions at 1% and the MET exon skipping at 1%. And the CNV was formulated so that it would be amplified. MET was amplified 1.4-fold over background. So that's the way we're able to report it from a liquid biopsy sample. You can see the sensitivity and specificity that we measured here in these columns. So again, very high sensitivity and very high specificity. So this is great, adding this new capability, but really wanted to understand kind of what is the lower limit of detection for these fusions. So what we did is take 5 different fusions that are measured and on our panel, targeted by our panel, and we measured the actual number of RNA transcripts in each of these materials by digital PCR. That's shown in this column here. And then we did a couple of libraries with this material, and we're able to detect all of these variants when you have about 10 or more copies of each fusion transcript in your cell-free material. So really able to go below the 1%, but another way of thinking of that is that we can accurately and consistently measure these fusions when they're present at 10 copies of the RNA or above. So that's another way of thinking about detection of these fusions in a cell-free nucleic acid from a liquid biopsy. Now, there's a lot of internal data that we generate when we launch a new product. We actually have a white paper that is describing this kind of second generation of assays. So there's this lung panel and the expanded variant detection, expanded classes of variants that are detected. And we really went through details about how we developed that capability and how we've tested internally. So this slide is just a teaser for you, really, to hopefully go to our website. Next week, this white paper should be available on the website. We talk about different blood collection tubes. We show results from EDTA and STREC. The EDTA, it really works well. It's that very widely available purple top tube, and as long as you're able to spin down the whole blood to isolate the plasma fraction, within 24 hours, that works very well and really provides clean results. We also have some information about STREC and different storage times in STREC. We have results from what the cell-free total nucleic acid looks like when you run that profile out on a bioanalyzer. It's kind of a new input type. Most people are familiar with cell-free DNA, so wanted to just provide information about what to expect. And here are some bioanalyzer profiles shown here. cfDNA from EDTA tubes on the top, and cfDNA from STREC tubes on the bottom. So hopefully I've given you a more broad overview of just our portfolio for the Oncomine cell-free assays. I've talked a lot about the library prep itself and mentioned that these are all optimized to sequence on our Ion Torrent sequencing systems. But I also wanted to point out, and the previous speaker did as well, that we have optimized analysis workflows that we build at the same time with the new library prep kit. So this is all working together, the molecular biology and the analysis, to really provide an optimized solution to be able to detect very rare variants from liquid biopsy samples with high confidence. We are, again, really testing down to 0.1% LOD, but you saw how that can alter a little bit depending on how much nucleic acid you have to put in. We tested down to the 0.1% limit of detection with 20 nanograms of input material. But the more you have, the better, right? It's really a dynamic assay that's able to detect, to use 1 nanogram of cell-free up to really as much as you have available. And then we've optimized content to include new variant types and to really focus on actionable mutations and resistance mutations. So on the next slides, I look at, and it's a slightly larger, Bea had a nice example of the analysis workflow. So I've got one here as well. This is actually looking at our Ion Reporter workflow for this Oncomine cell-free lung assay and showing the 3 variant classes. So this is the high-level report and visualization you get. CNVs and indels come first, and then CNVs, and then fusions. This is from our one of our positive controls that contains an EGFR and MET amplification. And the fusions over here are from our positive control fusion that has the EML4 ALK variant, an SLC ROS variant, and a CCDC6 RET variant, and also the MET exon skipping in here. So one thing you quickly see is each variant class has a QC test that is recognized by the analysis. So you quickly see that everything worked well. The QC passed, and if you don't have a variant or an example of one of these variants, it just says none detected, like the view you saw earlier. And if you have a variant that falls into that class detected, you see it here displayed. You can then click on these tabs up in the upper left corner to get more information about each variant class. So it's a really nice analysis solution. And here's another example from one of our customers in Spain that was able to detect a fusion in their cell-free total nucleic acid, and also an example of that in the solid tumor. So a couple of examples now that in this webinar of being able to detect these fusions from cell-free nucleic acid, and it's just a great capability that we've expanded our assays to be able to offer. All right, I think that's all I have, and I'll turn it back over to Christy, and I think we'll take some questions now.
Great. Thank you, Dr. Bramlett. Before we begin the Q&A, we'd like to ask attendees to give us feedback by taking our exit survey after the webinar. And for those who'd like to continue with our series on new frontiers in liquid biopsy research, the second part of the series will take place on April 26th. So we'll now start the Q&A. The first question is for Bea. Does the stability of cfDNA in liquid samples differ by sample type? For example, is cfDNA in plasma more stable than in urine, and so on?
I'm sorry, I have no experience with urine samples. Sorry about that. I can't help you there. Sorry.
Kelly, could you chime in on that one?
Yeah, we actually have some protocols for the MagMax cell-free DNA extraction that are specific to urine. Yes, you can get cell-free DNA out of urine. I'm not aware of extensive stability studies that have been done. It is slightly shorter in size. The fragments of cell-free DNA that you can get out of urine is slightly smaller than what you get out of plasma, but you certainly can use it in this type of assay and detect variants from it.
Great, thank you. Bea, could you please comment more on CNV fusion detection accuracy on real clinical samples? Have you done any internal validation?
For CNVs, we haven't detected yet any CNVs on the samples we analyzed. They were mostly patients, samples from patients with resistance to EGFR, but so far we haven't detected any MET copy number amplification there.
Great. And the next question is also for you. What kind of collection tube do you use? Is it an EDTA tube or another kind?
Normally we're using, yes, EDTA. In fact, all the results that I presented here today were done with cell-free DNA collected in EDTA. That's normally what we are using at our institution, mainly to keep it simple for everyone, because EDTA tubes are always available everywhere. However, I see no problem in working with other tubes stabilizing the sample, as our experience with other approaches such as digital or beaming, so on, they are working pretty well too. So if they are coming from outside the hospital, there should be no problem in principle on going on that.
Great. Kelly, the next question is for you, staying on the subject of tubes. If you've already collected a lot of plasma samples using Streck cfDNA tubes, can we still use these plasma for extraction of cfDNA using the MagMax cfDNA kit, or should the questioner newly collect samples using another type of blood collection tube?
I think you definitely can use the samples you have. Let me be clear about our experience with Streck. Really great option to add some room temperature stability if you're collecting in a different location. What we have seen is when blood has been stored in Strec, you can start to see DNA damage occurring in the cell-free DNA that can show up as an increased level of false positives. So what I would really recommend is using the material you have, and we actually have an optimized workflow for solid tumor, or FFPE, or really preserved, nucleic acid. And I would use that workflow if I'm using Strec material that has been stored for a long time. What that does is an optimized workflow for 0.5% LOD. And then you can use both analysis workflows and try to understand if you do have any of that, deamination event coming through. That's what we've seen from Strec tubes if the blood has been stored in them for a long time. But definitely it'll work, and I would say go ahead and use what you have to try to get as much information as you can out of it.
Kelly, how do you generate your reference material technically?
This S&V reference material, we actually take, we have a product called the Acrometrics Oncology Hotspot Control, and that's available online, and you can see a list of all the variants that are engineered into that material. I take a bulk from that, the manufacturing of that material, where all the variants are at 50%. I fragment it by sonication with a protectant in there to protect the DNA from damage during sonication. We then take the background material, a known genetic background, and we fragment that in the same method. And then we make mixtures of the two fragmented materials so that all of the engineered variants in the Acrometrics Oncology Hotspot Control material are either at 0.1% or 0.5%. And we verify all of that by digital PCR. I then use 6,000 copies of that fragmented control material into our testing that I showed the results of. And that's because it's a good estimate of how much DNA you would have in 20 nanograms of material. I think Bea, recommended or suggested about 5,000. We use 6,000, but yeah, it's right there in the range of what you would expect out of 20 nanograms of cell-free.
And do you believe engineered copy reference samples are enough to validate all the FFPE difficult sample conditions?
All the FFPE difficult sample conditions. I'm not sure I understand the question. I think it's just around the validity of these control materials. I've learned an awful lot about control materials. I think, in my experience, the types of control materials that perform the closest to actual cell-free nucleic acid are those that are derived from conditioned media, the way I described, for our CNVs and for our fusions. That really acts an awful lot like cell-free DNA or cell-free TNA. These other controlled materials, they do validate that you're able to detect the expected variants, but it's really nice to see in an actual clinical research sample like Bea showed here or I showed in one of my slides that you are able to detect these variant classes for sure. There is something lacking in our control materials that are widely available. And it's a limitation. What we do internally to try to get as close as we can to an actual clinical research sample is we test everything with blood that's been collected from healthy donors. So we really look at specificity from actual cell-free TNA or cell-free DNA that we've purchased from healthy donors. And that helps us keep track of how these assays and workflows perform on control material and actual cell-free TNA material.
Great, thank you. And Bea, how much bias is introduced by differences in amplification efficiencies of different variants?
What do you mean? Sorry, I didn't understand the question. We haven't tested for this. We haven't tested for this. No.
Okay. Kelly, is that something you can chime in on?
Yeah, you can get information from the analysis about the uniformity across the panel. So I would say, in general, we see about a twofold variation across the assays. So, for instance, when we're targeting 0.1% LOD, we really need 25,000 X coverage of each amplicon. In order to achieve that, we shoot for 40,000 X coverage when we're designing our experiment. So that additional sequencing coverage accounts for the variability that we normally see across all the amplicons from, variation in amplification efficiency.
Great, thank you very much, and I'm afraid that's all the time we have for today. We'd like to thank Bea Bellosillo and Kelly Bramlett and our sponsor, Thermo Fisher Scientific. If we didn't have time to get to your question, we will try to follow up with our experts. As a reminder, please look out for the pop-up survey after you log out to provide your feedback. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.